You will not understand. Actually, it is a net that is vasa recta. But in that, even though there are branches, messwork of capillary is here, in that also the main tube is two. One descending tube of vasa recta, and one descending tube of vasa recta. Similarly, we have one descending tube of the loop of Henle and the loop of Henle. You are able to understand? You are able to understand what I'm telling? You are able to understand? Okay. So we have ascending and descending limbs for both the vasa recta as well as the loop of Henle. But they are present counter to each other. Correct? Which is the ascending limb? The side vasa recta is the descending limb. The one which is the descending limb. Yes. The side vasa recta is ascending limb. Yes, so they are counter to each other. They are in opposite direction. Correct? Okay. Now coming to our point, that is how the concentration of urine takes place. Even though I taught yesterday, I will be again revising it because it is little confusing for you people to understand. That is quite normal. Every year it happens with me. Children, they don't understand at the first time. Okay. Now, for understanding this, we will consider something. Suppose if I say the previous filter, okay, the previous filter brings the sodium chloride here, then it will be very difficult for you to understand. Suppose if I say the tube is vacant, okay, the tube is vacant, nothing is there. Let us assume the tube is vacant, correct? Then what is happening? Now the total tube is vacant, nothing is there. This tube, this side, no sodium chloride, nothing is there. Then we will start from zero. Okay, we will start from zero. Everything is nothing, no sodium chloride here, no water release, nothing. We will start from zero now. Just to make you understand, I'm trying another trick. So the filtrate is coming here. There, the filtrate is coming through the PCT. What is the osmolarity here up to this?
300, ma'am. 300 osmolarity.
It is 300 milliosmoles. Okay,
Yes.
it is 300 milliosmoles per liter. That means per liter of the filtrate, what is the osmolarity of electrolytes? It is 300. Here, so 300 milliosmoles per liter of filtrate. Now, why it is exactly similar with the blood osmolarity? Because up to this, there is no change in the filtrate. So from here only all the changes will take place. Right? So all the changes will take place from this place. Because from here only the nutrients, some other electrolytes, some other ions are going in and out, but not exactly the salts. Right? So the electrolyte content is not disturbed up to this. So it is exactly the same as plasma. So filtrate is what? Excess amount of plasma containing everything, right? Containing everything. So that filtrate, which is coming through the Bowman's capsule, is exactly the same as our blood plasma, right? That is why the osmolarity is also same, because nothing has changed in that filtrate. No entry and exit of water and salt. So it was 300 in the blood. When it came out from the blood into the Bowman's capsule, then also it is 300. Because change has not taken place up to this. Next, what happens? After it flows little down, next what happens? So, let me show you here. Just a second. The movement is in this direction. Clear? When it reaches this part, so we will go up to this part. So this part water should be released. But now we will not talk about that. We will think that nothing is happening in this area and we have come here. We will start from the ascending limb. Okay, we will start from the ascending limb. Reason, the ascending limb releases salt, then only the descending limb will release the water. So first this has to take place. Clear? Okay. So now the filtrate which is going up, right? So that is releasing the electrolytes, the salt, the sodium chloride here. Okay, from the plasma. So all the sodium chloride are being released from the plasma. Now what is happening? As the sodium chloride is coming out, the salt part is coming out. So the the filtrate becomes more and more dilute. Clear? The filtrate becomes more and more dilute. Now, why more and more dilute? Because salts are coming out, means what is left in the plasma, the filtrate plasma? Only the water part. The more water, minimum salt. So that part is taking place here in the ascending limb. Now, how it is coming out? Why salt is coming out? Salt will come out by two processes. Okay. So, movement of salt from the ascending limb. Okay. Movement of salt from ascending limb takes place because of active and passive transport. Okay, active and passive transport. Now, active transport, we all know some energy will be spent and against the concentration gradient. Yeah? Against the concentration gradient means even though there are more and more salt in the interstitium surrounding tissue, still ATP will be used and some more salt can be thrown out of the ascending limb. That is called as the active transport. Passive means there will be channels, gates, protein gates. They will be sending the sodium chloride out. So both ways, the sodium chloride will be going out. Why? Because they have channels, gates for salts, electrolytes. But they don't have the channels, gates for the water. Okay. So the water gates are called as, this side I will write, and the water, is it visible? Water gates in the loop of Henle. Okay, I've used another pen, it's not visible, I think. The water gates in the loop of Henle are called the aquaporins. Aquaporins. Okay, so what are these aquaporins? These are protein molecules. Okay, aquaporins are the protein molecules. So these protein molecules on the membrane of this tubule cells, they act like gates. They will open and close so that the water will move. So which side aquaporin is there? I will put blue mark there. Okay. So this side aquaporins are there. Okay. So here you find the aquaporins on the membrane of, here, on the membrane of the cells of descending limb, but zero aquaporin on the ascending limb. So reason is aquaporin. Why it is impermeable? Why it is permeable to water? Because of presence of aquaporin. So this protein is present in the cell membrane of this tubular part, the cuboidal cells of the tubules. They have aquaporin protein, which acts like a carrier protein, which acts like a transporting protein. So that will help for movement of water molecule from tubule to outside so that it will be absorbed by the blood. But there is zero aquaporin on the ascending limb. So there is no water release from the ascending. Clear? So they don't have water release mechanism, but they have electrolyte release mechanism. So passive and active both transport. So this is passive and active, both transport that help for removal of sodium chloride out of the ascending limb. Clear? Okay. Now what
Yes,
is
ma'am.
happening? Now after the release of this sodium chloride, what is going to happen? What is the fate of this sodium chloride? Where is it going to go? So sodium chloride has several roles. This sodium chloride plays the most important role in the water reabsorption this side. Why? The question is why. Why we need sodium chloride, a salt this side for? Because it is permeable. Permeable means it will come out. Then why it is not coming out? If it is coming out all the time, then what is going to happen? The tubule will not have any control over the water reabsorption. So all the time will water go into the blood, right or wrong? All the time water, if there is no control mechanism, there is no stopping for water release from the descending. So even though the filtrate is there, even though a lot of water is there, still it needs a mechanism. Otherwise, what is going to happen? If there is no osmosis process, concentration gradient process, what is going to happen? Tell me. It will leak out all the water. So filtrate will always lack water. So what is going to happen? We will excrete highly concentrated urine, which is not desirable for our body. Right? That is a very bad condition. A highly concentrated. So that thick urine we cannot pass if there is no control mechanism for this. So what is the control mechanism? The control mechanism is osmosis process, right? So salt is required for the water to come out. Who is going to provide the salt? The salt will be provided by the same filtrate. You are able to understand? The salt is provided by the same filtrate, which is there in the filtrate, right? But that directly cannot come here because we have some stigma here, some stipulated work for the ascending and descending. This can release only water, this can release only salt. You are able to understand or not? You are able to understand? This can release,
Yes, ma'am.
release only water, this
Yes,
can
ma'am.
release salt. Right? But for this water, we need this salt to be present here. For water to come out, for release, this
Yes,
can
ma'am.
release, but the descending limb can release water. It is permeable to water. But what condition, at what condition it is removing the water? It needs a salty interstitium here. So the tissue which is surrounding this descending limb must be very salty. That means it must be hypertonic. Clear? So the outside I'm not able to zoom it. Okay, visible now?
Yes, ma'am.
Girls?
Yes, ma'am.
Okay. Let us continue. Okay. So, the salt is required for removal of water. Salt is there in the filtrate, but it cannot be removed here. Water can be removed here, but it needs salt. So they are all complementary to each other. So salt can be removed here, but that salt is required here. Right? You agree up to this? You agree up to this? You understand up to this? You understand up to this? Whatever I told now? I explained up to what? What are the things I explained? I explained about the location of the tube. I explained which is the descending and ascending limb, right? I explained about the vasa recta and its location. Correct or not? Right?
Yes,
Okay.
ma'am.
Now what else we explained? I explained which tube, which limb of loop of Henle is permeable to what? Correct? Up to that we discussed. Right? So this is permeable to water, this is permeable to salt. Right? But this water permeability, girls, just give me 1 minute. Just give me 1 minute. Is it visible? Is it visible? Is it visible?
Yes, ma'am.
I'm not able to hear you people. Any problem is there? Yes, Ashwini?
Ma'am. Visible, ma'am.
Your voice is not okay. I'm leaving and I'll rejoin.
Okay, ma'am.
Is it visible now? Is it visible?
Yes, ma'am. Visible, ma'am.
Okay, okay. So let us continue. I just got a phone call. Okay, let us continue. We were discussing up to the water is released dependent on the salt which is released on the other side. So this water will get released only and only if the salt is present this side. So this side has to be very salty, but the salt source is on the other side. So we need a carrier, a transporter who will bring the salt from this side to this. So nothing else can be a good transporter than our blood. Okay. So our blood will help also here for the transporting work, right? Okay. What else? What are the ways how the sodium chloride comes this side? Sodium chloride comes by two ways. One is the interstitium leaking through the tissue. Okay, so these are all interstitium. This, everything. It is packed. It is not hanging alone. It is not hanging freely. It is embedded, packed in the interstitium, tissue, inside the tissue. So through the leaking of tissue, salt will move from this side to this. Okay. What else method is there? The salt which is getting released from the ascending limb of loop of Henle will get immediately absorbed to the blood vessel, that is vasa recta. So majority of the salt will enter vasa recta and a minor amount will go through the tissues. But all are moving towards the interstitium which is near to the descending limb. Okay. So actually what happens, it is like a cycle, right? It is like a cycle. The salt which is released here and is coming here, right? So what is happening with the salt? Some of them will enter into the filtrate again after the water is being released. Salt is also being entering into the filtrate. Again, it is released here. Again, it is brought here. Again, it enters into the filter here. Right? You are able to understand what I'm telling? Salt release and like a cyclic process. The salt which is released here, that salt comes through the vasa recta here, released to the interstitium, and from here it helps for the reabsorption of water, bringing the water out of the filtrate, and it also enters into the nephron tubule. Why? Because again it has to create the same. It cannot be coming out, but it can enter into the filtrate. So some amount of salt enters to the descending limb also. Some they move through this interstitium this side. Yeah. So the one which is entering here, the same thing will be released here. Again, it is coming out, entering. Again, it is releasing on the other side. So there is a cycle of sodium chloride also. Being released, being absorbed again. You're able to understand? It is being released this side, coming this side through vasa recta, and from here it enters into the nephron tubule also. And by that time, it helps for removal of water from the filtrate. Clear? So, for understanding, we know that the nephron tubule is never empty, right? The nephron tubule is never empty. Moving through the vasa recta to this side. Okay. And the salt is released out from the vasa recta, from the blood vessel, and the tissue. Right? It needs osmosis process. And for the osmosis process, we need the salt. Right? The salt will be obtained in the other side. Now you are able to coordinate the things. Okay? Now you are able to understand a little bit clear what I'm telling.
Yes, ma'am.
If you are not clear, so you have to say, no, ma'am, we are not clear, so that I can teach again. If you don't understand also, you have to convey to me. You have to tell to me how I will understand. No also you have to say. Yes also you have to say. Right? Okay. So now we are clear about this. The salt and water concentration. Okay. So now the water is coming out. Right. As the water is coming out and some amount of sodium chloride is entering into the descending limb. So what is going to happen with the osmolarity? The osmolarity will increase or decrease. The amount of electrolytes will increase or decrease.
Increase, ma'am.
Water is coming out. Only it is electrolytes. Little water, more electrolytes. So osmolarity will increase. Correct? So from 300,
Yes.
as it moves down, As the filtrate moves down,
700.
because of release of water, because of entry of NaCl into the tubules, the osmolarity increases. So increasing to 600, 900, 1000, 1200. So by the time it comes this side. Why up to this? Because after this, it is impermeable to water. So water release happens only up to this. Clear? Okay, next what happens? After that, there is no water release. So whatever water was there in the filter, that is retained. Now only salt release. Okay, so the salt will get released. So osmolarity again, it reduces because salt is getting removed from the filter. So what is osmolarity? The amount of solute in the plasma. Clear? So that is osmolarity. So solute is moving out means definitely osmolarity is going to decrease. Is it clear? Okay. So osmolarity decreases, right? So up to here, when it reaches here around 300, dct around it is 270, 280, the osmolarity. Here, here also it again happens the same thing. Why now this part is over? We have understood about vasa recta and the loop of Henle. Now we'll come to the collecting duct. Okay. So now whatever salt was released here, the diluted, slightly diluted, now filtrate is moving through the DCT. Now it is coming again down via your collecting duct. Now collecting duct also comes under this gradation. Now we have to understand that also. How collecting duct from 300 it reaches the 1200. Clear or not? So that also we have to understand. Now come to this part. This part is around 280, 300, where the filtrate enters into the collecting duct. You are clear? Okay. Now the filtrate is coming down, right? So the filtrate is coming down here. It has entered and it is coming down. Is it clear? Now, when it comes down, this part has, this is interstitium. Okay, this part is interstitium. Now, what is present in this tissue? What is present? Why I gave this arrow mark? What is being released? What is being released?
NaCl, ma'am. NaCl.
It is NaCl. Now, I will put blue dots for NaCl. Okay. So this area is highly concentrated with NaCl. Right? So as the filtrate comes, is it visible? Children, is it visible? Is it visible to all?
No,
Not visible? Okay, just 1 minute. Wait for 1 minute. It might come, I think. Just wait for 1 minute. If it is visible, please inform me. Once it is visible, please inform me. Visible? My audio is coming? Is my audio coming?
Yes, ma'am.
Now it is visible. The board is visible. Is the board visible now? Board is visible or not? Kiran, my board is visible or not? Okay. Just a second, children. Is it visible, dear?
Yes, ma'am, but is not clear, ma'am.
Not clear. Okay. Kiran Mai is there? Kiran Mai, is it audible to you? Kiran Mai, is it audible to you? Okay, I'm making you the host. I will leave and I will again join. But if any problem is there, you have to allow me to enter. Okay. Okay. Otherwise, okay, okay. Is it visible now? Is it visible now, children?
Somewhat better, ma'am.
Somewhat better. Okay. Shall I bring it little? Shall I zoom in? Now, okay. Visible?
Yes, ma'am.
Okay. Now manageable. Okay.
Yes, ma'am.
Shall
Clear,
I
ma'am.
continue? Okay. Shall I continue now?
Yes, ma'am.
Yes, ma'am.
Okay. So we were in the area which is collecting duct, right? So the filtrate which is entering from DCT into the collecting duct, that is moving down to be excreted as urine, right? So the final phase of water reabsorption that takes place in this area. Okay, so the final phase of water reabsorption. So here some solutes were came out. The filtrate becomes slightly diluted, came here. Here what happens, the solute which came out from the filtrate, that will drag more water from the collecting duct. Okay, so what is that solute which came out from this tube, the ascending tube? The solute electrolyte is, which electrolyte came out? The sodium and chloride. Okay, the sodium and chloride, some will be entering into vasa recta for helping for water reabsorption from descending limb, and some part will remain in the tissue near to the collecting duct. Okay, so some amount of sodium chloride remains here. Some amount will enter vasa recta, that comes this side for water reabsorption. Some will move through the interstitium from this side to this. Okay, some will move here. Clear? So the sodium chloride present in the interstitium here near to the collecting duct will help for creating concentration gradient for the water, which is here inside the collecting duct. That means it is almost the urine. Now filtration part is over, reabsorption is over, water reabsorption is over. Now the substance which is coming through the collecting duct is almost the urine. Still, the final phase of water reabsorption will take place from collecting duct. With the help of what? I told, even though the cells are permeable to water, they will not leak the water. What you need for the release of water? You need a highly hypertonic environment. So the surrounding must be hypertonic so that the water will be released from the particular tube. Collecting duct is permeable to water, but it needs an hypertonic environment or hypertonic surrounding. So who is going to provide this hypertonic surrounding? The same sodium chloride, which was released from the ascending limb of loop of head. Is it clear? So sodium chloride, now which is present in this interstitium, helps for final phase of water reabsorption from the collecting duct. And at the end, the urine which goes to the ureter from here, that becomes highly and highly concentrated. You are able to understand now? Final phase of water reabsorption happens here in the collecting duct. Clear? And who is helping for that? The same sodium chloride, which was released here and which is helping for the water reabsorption from the descending. So what are the roles of sodium chloride? So sodium chloride plays two roles. One is water reabsorption from descending limb, okay, water reabsorption.
Ma'am, board
of
is not
descending
clear,
limb.
ma'am.
Board is not clear.
Yes, ma'am.
Okay, now okay.
No, ma'am.
No.
It's too
Anybody
difficult.
else who's not able to see the board?
Ma'am.
Okay, shall I erase everything? Now you people have understood this. Shall I erase and write what I'm writing?
Yes, ma'am.
Okay, I'm going to erase this. Anybody wants to understand anything from this drawing? Or I will erase it. Am I audible to you all?
Yes, ma'am.
Okay.
Erase it, ma'am.
So role of sodium chloride. Role of sodium chloride creates concentration gradient for descending limb of loop of Henle, and the second one is creates concentration gradient for the collecting duct. Okay, for the collecting duct. Okay, so this is the two roles played by sodium chloride. So very important this one. Not only it helps for water reabsorption from the descending limb, it also helps for the final water reabsorption from the collecting duct. Clear? So that salt electrolyte concentration helps for the final or maximum removal of water from the filtrate. So finally, the human being excretes a very concentrated, highly concentrated urine. Clear? So collecting duct, finally what happens? So one more thing, from the collecting duct, actually I erased the drawing, now I need the drawing. Okay. This part you people have written?
Yes, ma'am.
This part you have written?
Yes, ma'am.
I'm going to erase it. So here what happens? One more cycle happens here. Suppose this is our collecting duct. Okay. So from here water also gets released and small amount of urea from the filtrate also gets released. So here if our this is our loop of Henle. Okay, this is DCT. So this part, as I said, this is the thick segment, and there is a very thin segment before it becomes thicker, the ascending limb. Right? So what is this part? This is our ascending limb. Okay, it has two parts. One is the majority part, thick segment, and the small part is thin segment. Thick segment and thin segment. So from this point, it is ascending limb. From this point, it is ascending limb, but it has two segments. One is the thick segment, and the other one is thin segment. Clear? The urea which is released from here, small amount of urea that is also being released into the interstitial. So what is this tissue? This is interstitial tissue in which the structures are embedded. Actually, there is not this much of gap between the structures. The collecting duct is very close to the loop of Henle, and these are all packed like a coiled structure, all packed together. They're not very far away. These two tubes are also very close to each other. Clear? Okay. So now this urea which is coming here, that also creates concentration gradient. Right? So who are the electrolytes here? Sodium chloride and urea. So where does this urea come from? The urea comes from the filtrate. Why? Because our nitrogenous waste is urea. Ureotelic we are. Right? From that urea, small quantity comes out into the interstitium. So it adds up to the concentration grade. Clear? It adds up to the NaCl. So NaCl is released from the ascending limb. Urea comes from the collecting duct. Now all together they create hypertonic interstitium. So this tissue becomes hypertonic because of two major contributors. Now who are the major contributors? One is the sodium chloride released from this, okay, and the other is urea released from the collecting duct. Okay, so the urea which helps for water reabsorption after coming out from the collecting duct, again it enters into the thin segment of the ascending limb of loop of Henle. The urea comes out of the collecting duct, helps for making concentration gradient. That means it helps for making the hypertonic condition of the external part, that is the interstitial tissue. So creating hypertonic condition. Who are helping? Urea and sodium chloride. And after its job is over, like it's not required, the same some quantity will again enter here. Again goes through the filtrate. Again comes as a small quantity of urea continuously keeps circulating from the collecting duct to the interstitial tissue to the thin segment of ascending limb. Again going to the DCT, small amount keeps circulating. That is not excreted. So every time the filtrate comes, small amount will be coming out. Clear? So small amount, it is circulated. So this is called as urea cycle. Right? This is a cyclic process by which the urea comes out of the collecting duct, enters into this segment, again goes like a filtrate, again it comes. So it is keep circulating in the total this area of the tubules, the collecting duct as well as the loop of Henry. Okay, you're able to understand now? You're able to understand? Am I audible to you people or not? Am I audible to you people?
Yes.
Okay. Okay. Any doubt in this part? Concentrating urine counter current mechanism.
No, ma'am.
Any doubt? Okay. Shall we go to the next part?
Yes, ma'am.
Okay. So next part is regulation of kidney function. Regulation of kidney function. That means how the kidney functions, or we can say regulation of urine formation also. Kidney function, or we can say regulation of urine formation, or regulation of the nephron function. So all those mean the same, right? What are we going to discuss here? What are different factors which will control the formation of urine, concentration of urine, and related things? Yeah. So let us first go to various factors which control, regulate the kidney functions. In that, the first one, like different like hormone systems, we have the proteins involved in that, and we have brain involved in that. All things will be there. First one, what we are going to understand is regulation by ADH. What is ADH? ADH hormone. Okay, ADH only. Hormone is last H is hormone. What is the full form of ADH? Can anyone say? Can anybody say what is the full form of ADH? Am I audible to you people or not? If you don't know the answer also, you say we don't know, ma'am. Am I audible? voice is not audible. Okay. What shall I do now? Oh my God. Every time some problem is coming. Okay. Now is it audible? Is it audible now? Is it audible? Okay, I will. Am I audible? Am I audible? Is it audible, girls?
Yes, ma'am.
Okay. So I just asked one question. Can anybody say the full form of ADH?
Antidiuretic hormone.
Yes, it is called as antidiuretic hormone. Now, what is the meaning of antidiuretic? Diuretic means what? Diuretic urine means what? Diuretic means very diluted urine. Antidiuretic hormone means it will not allow the water to go out of our body through the urine. So when the body, it signals that the body is lacking water because of lot of urination, then we have certain receptors. So they will get activated and this particular hormone gets released. Then it acts on the certain parts of the neck of this dilute urine. How it will block? Directly, it will open some water channels, water gates, so that water. You're able to understand? You're able to understand? Okay. Now, which part of the body secretes this hormone? Tell me. 10th standard question I'm asking. 10th standard question I'm asking. Which part of the body secretes the antidiuretic hormone? Is it audible? Girls, is it audible? Is it audible to you all? Is it audible? Anybody can respond to me? Anyone can respond to me?
Yes, ma'am. It's audible, ma'am.
We'll do one thing. Shall we edit one Students, is it audible to you all? Children, is it audible to you all?
Yes, ma'am.
Should we stop today? Should we stop for today? Because a lot of internet problem I'm facing. Tomorrow shall we continue?
Okay.
Shall we continue?
Yes, ma'am.
Okay, ma'am.
Shall we continue tomorrow? Okay. Okay. Bye bye. Bye, children.
Bye, ma'am.
Continue tomorrow. Okay.
Bye,
The infection
ma'am.
of kidney. Okay. Thank you.
Okay,
Thank you.
ma'am.
Thank you, children.
Thank you, ma'am.
Okay. Thank you, children.